Good morning, everyone. Dr. Gribbins here. We are going to start recording lectures for Exam 1. And so we're going to start with Chapter 1 here. And Chapter 1 is more of a theme-based kind of introduce you to what we're going to talk about as far as large overall themes for the course. So I'm not going to cover all of Chapter 1. And the one thing that you need to realize is I'm going to give you a review sheet that will kind of cover the information that you need to know for the exam. And as you'll see when you go through the lecture, I won't cover every single aspect within each of the chapters. Some chapters I cover more of the information than others. And so today we're going to talk about some of the material that's in Chapter 1. And then our next lecture will be on Chapter 2, which is a review of chemistry, basically, or the chemistry of life. So let's dive in here and start talking about some of the overall themes. And so one of the overarching themes of biology is evolution, right? Or what we're going to term natural selection. And in Biology 155, if you've already had that, you've talked a lot about evolution, right? And diversity. If you haven't had 155 yet, you will talk a lot about diversity and how that diversity has arisen on our planet. And that is through the process of evolution or selection, right? And so organisms change over time. This change happens based on selection pressures on the selection process. And this is a core theme to biology because it's how organisms have diversified into all of the things that we see on the planet today. Biology itself, or the study of life, right? That's the kind of the general definition of biology. There are many questions you can ask, and we're going to, at the end of this lecture, talk about kind of scientific process. And then you're actually going to spend a lab or two on understanding the scientific process and how scientific method works and how scientific experimentation works. And we'll hopefully get some hands-on experience with that in the lab. But you can ask a number of questions, right? Biologists, like myself or like you, are very curious. We ask questions, right? We see or observe something in the environment. We ask a question about it, see if that question's been answered in the literature. If not, then we can come up with a educational guess or answer to that, which we call a hypothesis. And we'll talk more about that towards the end. So up here, I'm a herpetologist, so I study reptiles and amphibians. Those are my love. And here we have a snail-eating snake. There are snakes in the tropics that simply eat snails. That's all that they eat, and they've evolved to do that. They have very big heads. They're called blunt-headed snakes. And these snakes have very skinny bodies so that they can live arboreally or up in the trees. So as you can see here, this particular snake has found a snail. And basically, they have very, very sharp teeth so they can grab onto the shell, which can often be slippery, and then they eat the snails. And that's all they eat are snails. So it's pretty interesting. There are a whole group of snakes that have evolved over time to do this. Okay, so one of the big questions you have in biology, and one thing that you should be able to, as a biologist, explain is what is life, right? And it's a big question. And when you start thinking about it, you're like, I kind of know what life is, right? But when you try to explain it to somebody, it's kind of hard to understand or explain it to people out there that want to know what life is. So this picture down here shows a picture of Miller here in his lab in the 1950s and their Miller-Urey experiments, which happened in the early 50s. We're exploring how life arose on the planet, what we call the Big Bang Theory, and how we could make larger molecules or compounds from simpler elements and atoms, right? And so we'll talk about compounds, molecules, and atoms in the next chapter. But basically what they do is they apply heat and electricity to a primordial soup, and you can build membranes and other things, proteins, nucleic acids out of simpler compounds. So what we need to be able to do is kind of talk about what life is. And it's dependent on many specific but very identifiable things or substances, if you will. So this next slide kind of says, what's the difference between our dead plant here and our living plant, right? And the dead plant basically does nothing, but how is living being different than a dead plant that's doing nothing or is not alive? Living organisms, whether they're plants or animals, they have the ability to obtain and then utilize energy to build themselves, to organize themselves, to produce structure like the green leaves that you see in this plant. If I add water and good soil, this plant will continue to grow and add leaves as it captures the energy from the sun, right? And make sugars out of that using what we call carbon dioxide and water, and can then produce an energy molecule that it can utilize to run all of the processes in order that you see in a plant. So not only can they obtain and use energy, they use this energy for reproduction and growth. They use it to respond to the environment. That is, they have the ability, plants, if you watch them, can follow the sun. Their leaves kind of move with the sun so they can gather the most light. When it rains, the plants can sense rain or water and will absorb that through their roots. So they have the ability to respond to the environment. And they have the ability to regulate their internal environment, what's happening inside of them. For example, transporting glucose or obtaining water and holding that water in the cell and so forth. Also, over time, organisms that are alive, they adapt and evolve and maintain order, right? Maintain order. So let's go through a few slides to kind of talk about these four major kind of definitions of what we're terming life. So here we got energy processing. Organisms, like plants, are very important parts of the environment. So what plants do, they're called primary producers. Right? Plants are called primary producers. So a primary producer in our environment or in our niche or in our ecosystem, it has the ability to capture light, right? To grab energy, right? Environments or biological systems or open systems, they got to continually capture energy from our source, which is the sun. And then they fix that energy into an energy molecule. That is, they gather that energy and put it into glucose and other sugars that they then can utilize to build plant tissues, right? Those sugars are also the basis of, or backbone, if you will, of proteins. By adding some nitrogen to that and some phosphorus to that, we can make proteins and we can make nucleic acids. Other organisms will utilize plants, right? And they're secondary consumers. So we got primary versus secondary consumers. So secondary consumers, what they're going to do is then eat the plants or utilize products of the plants, right? So that's exactly here what our hummingbird is doing. This beautiful hummingbird from Costa Rica is feeding off of a flower of a particular plant. And what it's doing is gathering nectar, which is basically high in carbohydrates, sugars, right? Those sugars, the bird can then use and obtain an energy molecule, sugar, and then it's going to produce energy out of it. Cellular energy is made at the mitochondria. This is what this is, a picture of a mitochondria down here, right? As you can see, the mitochondria can take glucose and make ATP energy out of it, right? Things you've probably heard of before. So ATP is our energy molecule within the cell. So we got to gather energy. We got to continually eat, particularly birds and mammals like ourselves, because we maintain very high body temperatures. We have to eat a lot. And so we have to obtain nutrients from the environment and then transfer and utilize that nutrients into an energy molecule, ATP, that we can then use in the body. It's one thing that life does that non-living structures don't, like rocks or dead plants. We can take some of that energy and we can do reproduction, all right? So all organisms have to be able to reproduce. That is to produce a new organism, right? Offspring. So in this instance here, we're in Africa, we have giraffes, right? So we got a mother giraffe and her baby. And so, mammals can do this, birds can do this, reptiles can do this, protozoans, single-celled algae can do this. They can replicate themselves. They can produce either clones of themselves or they can participate in sex where they produce a new, unique individual, right? By mating with male and female. So reproduction is part of life. Some of the energy that is obtained from the environment is going to be utilized by the gonads to produce sex cells. Those sex cells are then utilized to make new organisms. Once we hatch from an egg, for example, here we got a crocodilian, then that 4-inch, 5-inch, 12-inch crocodilian grows, right? It grows into a 6, 8, 10, 12-foot organism over time. Growth and development are very important. So when an egg and sperm meet, they form a zygote, a single-celled organism that then divides rapidly, right, to produce new cells. And then those cells differentiate into all of the structures that we see in multicellular organisms. So not only do we reproduce, but we then utilize energy to grow and to produce many different types of cells that are organized into tissues and organs, which we're going to talk about that hierarchy of organization here in a few minutes. Also, organisms that are alive have the ability to respond to the environment. this rabbit down here, this hare is a desert-going species. It can basically see very well. It can see predators like large hawks, and hide from them. It also can help maintain or modify its internal environments. That is, its internal environment, the fluids, body fluids, and the cells and whatnot that are inside of the hare can actually respond to changes in the environment. With this hare, as the temperature warms up in the desert, they can send more blood to their ears. That's why they have such large ears, and you can see the blood vessels, right? You see the blood vessels in here that are going to be utilized to release heat to the environment so that they can cool their core. Here's a plant. This is a Venus flytrap that's actually captured an insect, right? That was attracted to the area, usually by some sugar secretion or something. And they land in the trap, and the trap responds by closing. So the plant actually can respond to the environment also. So this maintaining of a controlled environment internally, that is maintaining consistent conditions in your body, no matter whether the temperature goes up or down, or your oxygen levels change, or your salinity changes, or you eat and release things from the body. Mammals in particular, like this hare, we're strict what we call regulators. All right, so regulators versus conformers, all right? Something that you'll talk more about maybe if you ever were to take a physiology class with me. So regulators, like mammals and birds and the more advanced organisms like us, we maintain very specific conditions inside of our bodies, and we call that homeostasis. All right, so this process of homeostasis is the maintenance of controlled, consistent environment for our cells, that our cells are then happy, right? And they grow because and reproduce and live out their lives in a very controlled environment, no matter what's going on around the organism. That's called homeostasis, which means same environment or same conditions. That's what regulators do. Conformers, they kind of conform to the environment to maintain their homeostasis. So, for example, starfish or some of the other invertebrates that live in the oceans will have the same salinity inside of their body tissues as the ocean does. And that's kind of how they regulate what's going on in their body. Conformers don't necessarily always maintain consistent conditions within the body. They basically change with the environment. Regulators, on the other hand, are very, very keen, if you will, on regulating what's going on in the body no matter what's going on around them. So we maintain our body temperature 98.6 degrees no matter what's going on. And we do that because that's what our cells are happy at. They're happy at that temperature, and that's what homeostasis is. So up here I've got internal versus external, right? So organisms can respond to changes both externally, temperature, light, predation, and also internally. That is, you have kind of monitoring receptors or structures inside your body that can regulate your internal environment and what's going on there. And if something changes in your internal environment, you can respond to that change and reverse it or fix it. We're going to call those feedback loops eventually. So that's a little bit about responding to the inside and outside environments in the body. So living things can do that. They can respond to changes in the environment. And if you look around at living things, you see lots of order. You see lots of beauty in that order. And so this is the a macro shot of the inside of a sunflower, of the flower. And here are developing seeds. You can see how beautifully ordered that structure is. And there's a lot of energy that's obtained from the environment, the sun, that goes into that order. So organisms invest tremendous amounts of resources and energy to form their structure. Their structure is typically highly ordered. And that order fights what we call entropy, or the destruction of order, right? The second law of thermodynamics, right? Energy cannot be created or destroyed. It's simply transferred, right? That's the first law. And the second law is a lot of that energy escapes as heat or disorder. We maintain order by obtaining energy and maintaining the structure of our body and keeping things organized so that we can live out our daily lives, both internally and externally. All right, so that's a little bit about life, right? I've kind of mentioned how there's this hierarchy to biology, right? Or these levels of what are called organization. And I teach capstone, and this is one of the things that we kind of focus on in capstone, but I really try to focus on it throughout the courses that I teach because it's really, really important. Life can basically be studied at any of these levels of organization. The thing that's really interesting is if you go down all the way to the chemistry, which we're going to get into, right? And go all the way up to, say, biosphere, these are really different levels of organization or magnitude. Well, atoms, the smallest kind of units of matter that we're going to talk about, you can't see it. It's really hard to see even with the most advanced instrumentation, right? The biosphere, the planet, is huge. From atom to the earth are other levels of organization. Each level is going to interact and affect the level above or below it. And you can use lots of examples of this, right? So organisms, you can basically remove organisms from an ecosystem, and that's going to put a really large strain on the ecosystem and the food web, which can dramatically affect what the biosphere looks like, right? Or if you're more down here in the chemistry and molecular biology world, affecting what a molecule looks like structurally, changing its shape, can lead to pathologies, right? So a really good example of this, you change the shape of hemoglobin, which is a protein found in red blood cells, and you change one amino acid in that protein, hemoglobin, then you can get the disorder we call sickle cell anemia, right? You change the shape of the hemoglobin molecule, which changes the shape of the red blood cell, and then affects its ability to carry oxygen, right? So a lot of pathologies take advantage of structure, of function, at any one of these levels of organization. And so basically each level builds to make the next level above it. So the atoms that we find on this planet, which form the elements, right? Atoms make up elements. Elements can combine to form molecules and compounds. Molecules and compounds can combine to form really large, what we call polymers, right? The living polymers, the polymers that are really important for life are things like proteins, nucleic acids. So proteins, nucleic acids, fats, right? These are all examples of large macromolecules, right? These large macromolecules then combine to form your cells, right? And so a cell is the smallest unit of life that can do all of the processes of life, right? And cells are going to have, if you're a eukaryote like us, are going to have little compartments within them we call organelles. And these organelles are responsible for carrying out the daily activities or functions of cells. We combine cells to form the four tissues of the body. I'm also a histologist. I teach histology here. I really recommend you take it. Histology studies the tissue and their architecture within the body. There are only four tissue types in the body: epithelium, connective tissue, nervous tissue, and muscle tissue. So that's all we're made up of. Organs are combinations of those tissues, and then organs combine to form systems. All of the systems of the body form the organism. And if you affect an organ, that organ breaks down, that affects the organ system. The organ system doesn't work as well, the organism gets sick, right? So anything that you do to any one of these levels can affect the level above or below it. Different biologists study biology as it happens, life as it happens, at different levels of organization. So for me, I'm a guy down here. I typically am working in this area right here between tissues and organisms. I do all of those. There are other biologists in our department like Dr. Sweets and Dr. Milne who work with organisms, populations, and ecosystems. And then there are other biologists in our department that are molecular biologists, geneticists, people who study cancers and they work in the molecular realm. Most biologists these days are integrative. They work at more than one level of organization and to do the research that they do. So biology is an integrated science. So I give you a couple pictures here on the bottom. Here is the chemical structure, if you will, or the atomic structure of chlorophyll. Chlorophyll, very important pigment, right? Well, it has certain atoms that make it up. Particularly carbon, oxygen, nitrogen, and phosphorus are going to be found in a lot of these large molecules. And what chlorophyll is, it's a pigment. It can absorb light, right? And the chlorophylls are produced in an organelle, which is a membrane-bound structure inside of specific cells within the leaves of plants, for example, or within the cytoplasm of algae. And these are called chloroplasts. And chloroplasts, you can see, have all of these stacks of membranes within them. And within those membranes are going to be chlorophyll molecules. And what they do is they capture light. So as light comes down and hits, as you can see with my laser, hits the chloroplast, the chlorophyll absorbs that light. And that is going to affect the atom relationship to one another. Electrons are going to move around with the absorption of that light. And we're going to use that energy to split water, right? And then we're going to talk about photosynthesis this semester. The water that's split, the plant releases oxygen. It grabs up the protons, or what we call hydrogen atoms, and then we'll basically reduce carbon dioxide, that is, add the hydrogens, that's what reduction is, the hydrogens to carbon dioxide to make carbohydrates. And so this is what a plant cell looks like here. So this is the entire cell. The little green circles are the chloroplasts. So this is a light microscope image all the way on my right here, if you're following my pointer. This is an electron micrograph from a transmission electron microscope that shows you a chloroplast. This is obviously highly magnified, right? So here's a 1 micron bar to show you that chloroplasts are about 3 to 5 microns in width. Or this cell here, this plant cell, is probably close to 50 to 60 microns in width. And then the individual chlorophyll molecules are inserted into those membranes within the chloroplast, and their job is to gather light. So using that information, I'm kind of combining all of these levels. Obviously, the cells here are going to be found in leaves, and leaves have a particular structure, and the cells that do most of photosynthesis in a plant, a higher plant, are going to be in the leaves. Okay, so this is an image from your text that kind of shows you all of these linkages using pictures. So you got molecules here that are going to combine into basically what we call macromolecules, which then combine into organelles, and organelles form cells. Cells are surrounded by membranes. These cells have particular shapes and structures, and they are going to be organized into tissues. Here in the plant, we have cells that are organized into the tissues of the leaf. Here's the organ, the leaf, and then the leaf is part of the organism, which is a tree, right? And these trees are part of a population that are found in a community and ecosystem, which is part of our biosphere. So all these levels are linked to one another. Okay, another major theme that I want to talk about. So we've talked kind of about evolution, right? Mentioned it. Talked about what life is and the processes of life, right? We talked about the organization hierarchy that you find at different levels of organization within the biological realm. And within that, another really, really major thing is as an anatomist, as a histologist, anatomist, and I also teach physiology, is the importance of structure and the relationship of structure to function. So what something looks like often in the biological world is going to determine its function. That's why I'm a histologist. What you see determines how something works. And it's like looking at a picture book, and that's what a histologist does. That's what an anatomist does, right? And so structure and function are very important at all the levels of organization. So if you're looking at a bird, like here, this is a seagull. The seagull can fly because of the way it's put together, the way it's built, what its structure looks like. It has wings, right? These birds are also very light because their bone, this is a picture of the bone here, from the arm of a bird, is highly pneumatized, all right? So it's a word you may not have heard of before. I'm just going to pneumatized, which simply means that it has lots of airspace. Pneumonia, the pneuma, that part of Greek means airspace. So pneumatized bone it is going to have a lot of airspace in it. It's got a lot more airspace and a lot less bone than you would see, for example, in our arm. Therefore, the bone is much lighter, right? And so the bird can fly. So structure equals function. The ability of the bone to look the way it does, the architecture of the bone helps determine what the animal can do. Here is flight. Now that's at the organismal level. You can look at the structure of a macromolecule and determine what its function is. You can look at the structure of a cell and see what it does, or determine that which helps determine what its function is. Nerves have really long cellular processes to conduct electricity to communicate quickly within our bodies, for example. So there are lots of examples of this. We'll talk a lot about structure function. As an anatomist, I teach a lot of structure function. I also, as a physiologist, teach the structure first in order for you to understand the function of what things do. So structure and function of living organisms are really closely related. There are millions of examples, if you will, of how structure determines function, and we'll get into a lot of those. So the structure function kind of theme is going to be very important in all biological classes that you might take here at UIndy. So as I mentioned, this happens at many levels of organization. So you can look at the structure of atoms, you can look at the structures of cells, you can look at the structure or what an organism looks like, how it's put together. All of those will affect the function and organization and what activities those particular organisms can do. The cell is the lowest level of organization that performs all the activities of life, right? So all of those things we talked about, cells can obtain energy. They can then use that energy to perform work by producing ATP or capturing ATP, right? Or making ATP. They can grow. They can divide. They can reproduce, right? They can control the internal environment inside of themselves, inside the compartments we call organelles. They can do all of those things that we've talked about. All cells are going to be enclosed by membranes, and all cells are going to use DNA as their genetic information. So what does that mean, as their genetic information? That means as basically their instructions, their blueprint of how to stay alive, of how to be alive, how to obtain what we call life. And so the genetic information is a code, and that code is utilized to maintain the everyday activities in life of a cell. Eukaryotic cells, or most of the cells we're going to talk about in this, in this section of freshman biology, or what we call introductory biology, are going to be membrane-bound cells. They have a cell membrane, but also inside the cytoplasm is compartmentalized or separated into compartments. Those compartments are also membrane-bound, and we call those organelles. So things like mitochondria, nuclei, rough endoplasmic reticulum, Golgi apparati, vacuoles, vesicles, these are all membrane-bound organelles. By comparison, smaller, simpler cells called prokaryotic cells, they still have a cell membrane that surrounds them, but they have no real compartmentalization of their cytoplasm. Their DNA is also typically circular, all right? Whereas eukaryotic cells, their DNA is typically linear. And prokaryotic cells do have ribosomes, as do eukaryotic cells. Ribosomes look a little differently, and the ribosomes are a little bit smaller in prokaryotes. We're going to spend a lot of time talking about eukaryotic cells. We're going to basically work our way up to eukaryotic cells and talking about some of the processes that they do. This is a picture. This is an electron micrograph again of two types of cells. This is a eukaryotic cell here, obviously, right? If you're following the pointer all the way around, and here's a prokaryote. This is a bacillus bacteria or a rod-shaped bacteria, and you can see the size difference, right? So prokaryotes are much smaller. For the most part. And you can see they have no nucleus, they have some DNA. The DNA is really sitting in the middle of the cell, and then there's no membrane-bound organelles. When you look at a eukaryotic cell, we have a really big compartment here called the nucleus. That's where all of our DNA is. We also have other membrane-bound structures out here, like vesicles and mitochondria and chloroplast and endoplasmic reticulum in blue here, that are responsible for doing certain functions of the cell. And the reason they can do those certain functions is because of what they look like, the membrane-bound structure, and also because they compartmentalize or separate their interior versus the rest of the cytoplasm. So compartmentalization becomes really important. The ability to segregate or isolate areas or compartments within cells and within the body become very important in the physiology of organisms. And membranes play a major role in that. But as I've already mentioned, DNA is really important, right? DNA and RNA are really important. So chromosomes contain most of the cellular genetic material. Not all of it, but most of it, right? And the chromosomes are made up of DNA. So you got a circular chromosome, typically a single circular chromosome in a prokaryote, and then you can have multiple chromosomes in eukaryotes. So as you all know, humans have, 46 chromosomes, right? These are long linear pieces of DNA. There's lots of it. You have several feet of DNA in each of the individual cells that make up our bodies and our organs. DNA is deoxyribonucleic acid, and DNA is the substance of what we call genes, or is made up of genes. And what genes are long sequences or codes that basically are going to read or code for something, right? Typically they code for proteins. The genes are also units of inheritance. We have to pass our whole book, if you will, all of our instructions, which is what the DNA are for the cells, to other cells if we undergo mitosis or meiosis. If an egg and sperm meet, we got to make sure that the egg and sperm, when they come together to form the zygote, contain the entire book, if you will, or all of the genetic material or DNA of that particular organism. So that's why sperm and egg and humans each have 23 chromosomes. So when they come together, you get the whole book, 46 chromosomes right together. So the ability of cells to divide, the ability to copy this DNA and then to pass it to the daughter cells is really important. Every cell in your body has the same book, but not every cell in your body uses the whole book, if you will. All right, so liver cells use certain pages in that book. Muscle cells use certain pages in that book. But each cell, whether it's muscle, nerve, liver, connective tissue cells, all have the entire genetic code, if you will, of the organism. So these are in eukaryotic cells. The DNA is going to be contained in the nucleus, right? And so down here you have two pictures here. Whoops, sorry. Go back. Hopefully. Oops, going the wrong way. Sorry. Down here you can see the cell here. This is a cell that's not dividing. This is the nucleus in the middle. And we have labeled some of the components of the DNA, and it turns the DNA blue. This is a cell starting to divide. So you get the green here are microtubules, what we call the spindle apparatus. Over here you got chromosomes separating, and you got the spindle apparatus in the middle. This is what we call anaphase of mitosis. And we're basically copied the genetic code, and we're separating it into two cells so that each cell receives a complete copy of the DNA. So each chromosome is a long DNA molecule with hundreds of thousands of genes. Genes are a code that can be read, or information that are going to code for proteins. So the genes typically are going to regulate protein production, or what we call gene expression, and proteins in the cell are what run kind of the everyday activities, or what we call the physiology of the cell. So a lot of proteins, for example, are enzymes, right? And enzymes are going to be proteins that are going to run chemistry, if you will, or chemical reactions within the body. And so DNA is inherited by offspring from their parents. So egg and sperm come together in a human, form a zygote, right? So you got 23 chromosomes in the sperm, 23 chromosomes in the egg. They come together to form a zygote, a fertilized egg that has 46 chromosomes. We then divide that cytoplasm up into hundreds of cells, and then those cells divide further and differentiate and eventually form all the organs and structures of a baby, right, of the offspring. As the offspring grows, which is one aspect of life, and differentiates into all the organs, we then get the whole organism. Okay, so this is what DNA looks like, right? So you can see this is the double helix that we have of DNA. So DNA is a double helix, and it's basically twisted around each other, and the two strands. The DNA is made up of a backbone here, as you can see, that is made up of blue and yellow pieces, right? Which is basically a sugar and a phosphate group. So the blue is represents your phosphate. Is your sugar group, what we call deoxyribose, all right? Spell it right for you. The deoxyribose, right? And then the yellow is our phosphate groups. So we got a sugar backbone, right? That's made up of, that has phosphates with it. And then coming off of the sugar backbone, as you can see, are what we call nucleotides, right? And so these nucleotides, there are 4 of them, A, C, T, and G, are going to be arranged in a sequence. Let's say this piece of DNA right here that we have represents a gene. So this whole thing is a gene, right? And so what a gene is simply a sequence of DNA, a strand of DNA that has a sequence of bases in a particular order that then codes for amino acids. And so we bring in amino acids based on this code. All right, so these are the instructions, if you will, for this gene. All right, and this gene is this many bases long. All right, and you can see it's got A, C, T, A, T, A. That is the unique sequence of bases that are lined up. That's the code. And this code is going to then be read by ribosomes. All right, those ribosomes will read the code and then produce the right protein. The protein is basically a long strand of amino acids. So we bring in the right sequence of amino acids. That protein will then take on a particular three-dimensional shape in the cell based on its amino acid sequence. And then that protein will perform a function. What I'm giving you is basically what we call the central dogma of molecular biology, which is this process right here. DNA can be read and then copied into a single strand, because remember, DNA is double-stranded, right? Can be copied into a single strand, an exact copy we call RNA. That RNA, which are instructions, can then leave the nucleus and go out to the cytoplasm and find a ribosome. And at the ribosome can read the RNA, the code, and produce the proper sequence of amino acids, which we call a polypeptide or a protein. That protein then can take on three-dimensional shape in the cytoplasm and do function. So this process of RNA to DNA, which is called transcription, to transcribe or copy, that's what that means, to transcribe our gene of interest into RNA. That RNA then goes out to the ribosome, right? So this happens here, all right? This, what we call translation, happens in the circle here, which is the ribosome, right? The ribosome reads the RNA and then builds the protein from the instructions. So it takes the instructions, reads the instructions, builds the protein, right? That protein then takes on a particular three-dimensional shape, its structure, which then performs certain functions. The unique sequence of bases, which give you a unique sequence of amino acids, is what's responsible for producing the shape of the protein, the structure. That's how you have all these different enzymes that look different from one another, because their sequence of amino acids is different based on the unique sequence of the code that is found within the bases of genes or pieces or strands of DNA. This process here of DNA, RNA to protein is called gene expression. Right? It's something you'll learn in molecular biology. This is a major thing we're going to talk about both in this course and as you go along and take other biology courses at UIndy. We're going to talk more about this as we get past the chemistry level of organization, and we're going to talk a lot about central dogma and how it works, and then get into some more molecular biology as we progress through this course. Okay, the last thing I'm really going to talk about today is just introduce you to the scientific method. So the scientific method, we're going to do a bunch of labs on the scientific method and utilizing the scientific method, and then writing based on what we've done in lab, and what we're going to do in lab is based on the scientific method. So this kind of lists kind of the ways a scientist utilizes science, if you will, or experimentation to test observations that are made out in our world. So the first thing that biologists are very curious, and they are very keen. They look around, they see things happening in the environment. Say if we're a biologist or out in the world, they make observations. These observations lead to questions, right? The questions we can then answer educationally, or make a guess, if you will, which is called a hypothesis. Now, the one major thing with a hypothesis that's very important, it has to be testable. That is, I have to be able to, in this natural world that we live in, make an experiment or test that answer to see if it's actually right. If it's not testable in the realm of the natural world and biology, then it's not a good hypothesis. It's not a hypothesis that we can use in a biological sense. But if I have a nice, simple, testable explanation for what I'm observing, then I can build an experiment based on my hypothesis, right? Typically, the hypothesis leads into a prediction. It's an if-then statement based on my answer to the question or the answer I think is to the question that I observed out in the environment. These predictions can then allow me to build experiments. Experiments are a set of controlled steps that allow me to test whether my hypothesis is right or not, or whether it answers my question or not. Results are data that I collect from the experiment, and then I can use these results to interpret whether my experiment actually helped me answer my question. So I can accept the hypothesis that, oh yeah, that was a good guess, or I can reject the hypothesis. When I reject the hypothesis, maybe it'll lead me to other questions that I can provide other answers for, and then I can test those hypotheses over time. So the exploratory and discovery part of this is observing nature and asking questions, and then reading the scientific literature. So if you're interested in a particular facet of biology, let's say sperm production, for example, which is something that I study in reptiles, you could go out and read all of the literature on sperm production in reptiles. If you read that literature, then you get a foundation for how that particular avenue of research works, right? And so if I find a lizard out there in the world that I want to study the reproductive behavior, or I want to study sperm production in, I can utilize that foundation to build an experiment and come up with a hypothesis about how that lizard might fit into how the rest of the lizards reproduce, for example. We're going to practice doing all of these pieces as we go through the lab, right? The lab sequence of this course. So an experiment, you have to have a few features to make up a good experimental design. And so you got this little image up here that shows beakers and whatnot if we're studying chemistry. But all experiments that we do in biology typically have to involve variables, right, and controls. That way we know that what happens when we manipulate a particular variable within our experimental design, the controls help us see if those variables are what are actually doing the changes that we're seeing in our experiment. So if you're looking at a good experimental design, controls provide a baseline or a standard. Whereas the variables, there are two major variables. There are what are called the dependent variable and the independent variable. The dependent variable is what we're measuring. It is dependent on how we manipulate the independent variable. The independent variable is not dependent on anything. It's what we actually manipulate or change in the experiment. The way I remember it is dependent is what we measure, independent is what we manipulate. So dependent is what we measure, independent is what we manipulate. So let me give you an example, right? Because these are definitions. So if I was interested in seeing if a fertilizer causes growth, right? Increasing growth in, say, corn plants. The way I would set this experiment up to get a good experimental design is that I would have a group of corn plants that I would call the controls. And I would give them just water and keep everything else the same amount of light, the same amount of soil, the same amount of light and dark that they are exposed to. And then I would take my experimental group, which is another set of corn plants, and I would add the fertilizer to the water, right? And then add that to the soil. These plants that have the fertilizer in them are going to have the same amount of soil. They're going to be exposed to the same amount of light as the control. So everything's held consistent except you have fertilizer added to the water in one set of plants and just water in the other set of plants. The plants that receive just water are called my controls. They'll give me a baseline of growth, how much growth actually happens with no fertilizer, right? It's called a negative control. Whereas my plants that are the experimental, I've added fertilizer to, that's my independent variable, right? What I've changed is adding fertilizer. The dependent variable I'm measuring is growth. Does that increase growth, adding the fertilizer? Does it decrease growth, or does it not change at all compared to the controls? So that's how we use controls, dependent and independent variables. And obviously experimental replication is important. We don't want to use one corn plant for each of our control groups and our experimental groups. We want to use 10, 20, hundreds of corn plants. If they all respond the same way, then you can be pretty confident that changing, for example, fertilizer causes an increase in growth. If it only causes an increase in growth in one plant, that might not be very convincing, right? If we have changes in 100 plants that are exactly the same, that's pretty convincing that what we're seeing is not just by chance alone, right? If 100 plants are growing larger than the controls, which are another 100 plants, then we feel more confident that it's actually the change, the actual dependent variable that's causing this change. It's not chance, a chance event. So that's why replication is so important. Okay, I think that's it. All right, so that'll be the end of lecture 1. I will, on Monday, put up lecture 2, and that's going to be a review of chemistry. All right, so I hope you have a great day.